Imagine a patient walks into a clinic, and they're suffering from severe progressive vision loss, something called cone rod dystrophy.
Which is devastating on its own.
Right. But then the doctor examines them, and they notice something completely unrelated. The patient has absolutely no tooth enamel.
Wow. Amelogenesis imperfecta.
Exactly. So what could possibly connect the eyes and the teeth?
It honestly sounds like the setup to a medical riddle, but I mean, it's a very real condition.
Yeah, it's called Jalili syndrome. And the mystery deepens, because in other cases with similar underlying issues, patients present with profound developmental seizures or even severe metabolic failure.
Which makes the diagnostic landscape completely murky for doctors.
Totally murky. But it turns out the thread connecting the eyes, the teeth, the brain, and the metabolism, it's a failure in how the body handles a single, seemingly basic element, magnesium.
Which is incredibly easy to overlook, right? I mean, we talk about calcium for bones, iron for blood, but magnesium is really the unsung hero of systemic cellular biology.
It really is. And specifically, these devastating diseases are driven by mutations in a family of proteins that act as like molecular bouncers for magnesium. They're called CNNM proteins.
Right, the bouncers that control what gets in and out.
Exactly. So today's deep dive is going to look at the exact mechanical blueprint of how these bouncers work. We're breaking down a fascinating 2026 paper published in the journal Cell by Bai and colleagues.
Yeah, it's a massive breakthrough. For the first time, researchers actually managed to freeze and capture the complete 3D structural blueprint of the full-length human CNNN4 protein.
And they did this using a technique called cryo-EM, right?
Yeah, cryoelectron microscopy. Basically, they flash freeze these proteins in this possibly thin layer of ice, and then they hit them with a beam of electrons to take thousands of two-dimensional pictures.
And then computers stitch all that together.
Exactly. The computers stitch it into a highly detailed 3D map. It allows us to literally see this microscopic machine atom by atom.
That is mind-blowing. And look, if you're listening right now, this isn't just abstract textbook biology. Magnesium is vital for your energy, your brain function, your metabolism.
Oh, absolutely.
We are zooming in on a highly complex machine operating inside your cellular membranes right at this very second. And, well, what the researchers found when they developed this 3D map was completely unexpected.
It really was. Up until this paper, science mostly relied on the blueprints of similar proteins found in bacteria.
Because they're easier to study, I assume.
Yeah, much easier. But the human CNNM4 structure revealed an architecture and an operating mechanism that is, well, wildly different and vastly more complex.
So let's build this machine from the ground up, or I guess from the top down. To visualize the structure of human CNNM4, I want to use a specific mental image for you guys. Picture a 4-story building. Let's start up at the roof. The top floor.
So that top floor is known as the extracellular domain, or ECD. And as the name suggests, this part sits entirely outside the cell. It just floats out there in the extracellular space.
Okay, so it's outside. What does it look like?
The standout feature is that it's heavily decorated with complex sugar molecules. In biochemistry, we call this being glycosylated.
So it's like the roof is covered in specialized antennas or ID tags.
That's a great way to think about it. The cell attaches these sugar tags to specific amino acids to traffic the protein. It's basically the cellular shipping system.
Ensuring this complex building gets transported to the right neighborhood.
Right, specifically to the outer plasma membrane of the cell.
Okay, so from the roof, we take the elevator down to the membrane itself. The elevator shaft is the transmembrane domain, the TMD. This is the part that actually spans the barrier between the outside and the inside of the cell.
But for a transporter protein, it's incredibly tiny.
Yeah, that surprised me.
It's totally minimalist by design. It crosses the cellular membrane using only 3 helical structures per protein subunit. Usually, transporters have massive, bulky domains crossing the membrane, but this one is really streamlined.
Then we drop down inside the cell to the first basement. This is the energy center, officially called the CBS pair domain.
And below that, anchoring the whole thing, is the deep basement, the CNBH domain.
Right, so we have our 4 floors, the extracellular roof, the transmembrane elevator, the energy center in the first basement, and the deep basement at the very bottom.
Now, here is where the structural reveal gets fascinating. Because in simple bacteria, 2 of these protein subunits just pair up perfectly face to face.
Like a mirror image.
Exactly. They form a simple, symmetrical pair. Picture 2 people standing straight up, looking right at each other, mirroring each other's movements.
But the human version doesn't do that.
Not at all. The researchers found that human CNNM4, forms what they call a dimer of asymmetric dimers.
A dimer of asymmetric dimers. Okay, break that down.
You have 4 identical protein subunits coming together to build this machine. They pair up, but instead of mirroring each other perfectly, they contort into completely different shapes, and they actually swap domains with each other.
It's like instead of standing face to face, the 4 subunits are crossing their arms to hold hands with the person next to them, creating this interwoven knotted chain.
Yes, a very tight molecular knot.
But wait, I'm stuck on something here. If the human body is building these 4 subunits from the exact same genetic sequence, why wouldn't they just line up symmetrically? Making them cross arms and change shape seems like a massive waste of cellular energy. Why make it so complicated?
Well, it's a trade-off.
Yeah.
You're trading energetic efficiency for absolute control.
Control? How so?
Those extra structural floors,
Yeah.
the massive roof, and the deep basement are unique to complex organisms like us. Bacteria just don't have them.
Ah, okay.
So by forcing the protein to domain swap and create this asymmetric structure, the cell physically wires the transport units together.
Yeah.
They aren't just sitting next to each other. They are mechanically linked at the top and the bottom, creating a massive communication network.
Oh, I see. It allows the human cell to regulate the transporter with a level of nuance that a bacterium doesn't need. It's essentially cellular middle management.
Precisely. It allows the body to integrate multiple signals before deciding whether to open or close the magnesium gate.
Okay, let me pause and make sure I'm tracking this blueprint and give you guys listening a quick recap. So bullet 1, we've got a 4-story machine, the ECD roof outside, the TMD elevator, the CBSP energy center, and the CNBH deep basement.
Spot on.
Bullet 2, it's an asymmetric tetramer. And bullet 3, instead of simple symmetry, they domain swap to create complex regulation.
A perfect summary.
So for you listening, I want you to ask yourself, have you ever noticed how more complex systems in your life require more middle management? How do you think this microscopic machine coordinates all these moving parts without just, falling apart?
Which brings us down to the first basement, the energy center. Because this machine doesn't just run on good vibes.
Definitely not.
It needs a very specific fuel to lock together. And the researchers noticed something fascinating about how this machine turns on. It requires a molecule called MgATP, magnesium ATP.
ATP being the fundamental energy currency of your cells, right?
Exactly. But here's the detail that caught the researchers' attention. If you expose the protein to ADP or AMP, which are basically just ATP molecules with fewer phosphate groups attached, the machine absolutely refuses to turn on.
It just sits there.
Yeah, the subunits just flop around dynamically.
Yeah.
The body strictly demands ATP.
Why is it so picky? I mean, ADP is almost the same thing.
It all comes down to reach and repulsion. See, ATP has 3 phosphate groups forming a long tail. ADP only has 2.
Okay.
That third phosphate on ATP makes the tail just long enough to physically reach across the microscopic gap between 2 interwoven protein subunits.
Ah, to bridge the gap. But there's a catch, right?
Yeah.
Because phosphates carry a heavy negative electrical charge.
A very heavy negative charge. So if you have 2 ATP molecules reaching across the divide toward each other, their negative tails repel each other violently.
Right. It's like trying to force the negative poles of 2 strong magnets together. They'll just push each other apart. So how does the protein actually lock together if the fuel repels itself?
That is where the magnesium part of MgATP comes into play. The cryo-EM map revealed a single specific magnesium ion sitting perfectly in the crossfire between those repelling APP tails.
Oh, wow.
Yeah. And magnesium carries a double positive charge. So it sits there acting as a molecular salt bridge. It neutralizes the negative repulsion and physically cements the 2 sides together.
So without that third phosphate, if you only have ADP, the tail is too short, no bridge forms, and the machine falls apart.
Exactly.
That is just incredible biological engineering. But there's another layer to this basement. Just above this ATP ignition switch sits a physical cluster of amino acids called the acidic patch.
Yes. This patch is rich in specific acidic amino acids like E497 and D498. And chemically speaking, they carry a negative charge.
Which means they attract positive things.
Right. Because they are negative, they act like an electrostatic magnet for any free-floating, positively charged magnesium ions in the surrounding cellular fluid.
And this connects right back to our hook at the start. When we talked about Jalili syndrome, the vision loss, the lack of tooth enamel, we mentioned it was a genetic mutation. That mutation happens right here.
specifically at residue D498. Yeah.
A single amino acid in this acidic patch is swapped out incorrectly due to a genetic error. It's a literal hardware glitch in this exact sensor.
It really emphasizes how critical this tiny patch is to human health. The researchers hypothesized that while the ATP site acts as an on and off switch, the acidic patch acts as a sensor. It literally tastes the fluid inside the cell to see how much extra magnesium is floating around.
Okay, let me try a car analogy to see if I have the mechanism right. The MgATP salt bridge, where the magnesium locks the repelling ATP tails together, that's the ignition key.
Yep.
You absolutely must have it to turn the engine on and lock the structure in place. But the acidic patch sitting just above it, that's the gas pedal.
I like that.
By sensing the ambient magnesium in the cell, it tunes the speed of the transporter engine up or down, depending on what the body needs.
That's highly accurate. The cell is using the local concentration of the very element it transports to regulate how fast the transporter operates.
So summarizing the energy side of things for the listeners with a quick recap. Bullet 1, MgATP cements the structure via a magnesium salt bridge. Bullet 2, the acidic patch acts as a cellular magnesium sensor. And bullet 3, mutations here cause the devastating effects of Jalili syndrome.
Perfect.
So think about your own body's energy levels for a second. How does your body know when to conserve versus when to expend? It's wild to think this protein has a built-in atomic sensor to do exactly that at a microscopic scale.
It really is. And once that sensor says go, the actual heavy lifting happens, the physical movement of magnesium out of the cell.
Which takes us back up to the elevator shaft, the tiny transmembrane domain. The researchers note that CNNM4 is an antiporter.
Right. It doesn't just pump magnesium out for free. It operates on a strict exchange system.
Oh.
For every magnesium ion it pushes out of the cell, it allows a sodium ion to flow into the cell.
So the inward rush of sodium provides the mechanical force to eject the magnesium. Magnesium out, sodium in.
Exactly.
But to actually see this exchange happening, the researchers had a massive problem. This machine is dynamic, right? It's constantly moving. To understand it, they needed to take a 3D picture of it in different poses.
Yeah, they easily caught it in an outward-facing pose where the channel is open to the outside.
Yeah.
But they desperately needed to see it in the occluded state.
The occluded state meaning where the channel is sealed shut on both ends with the ion trapped in the middle. But how do you take a picture of a moving elevator while the doors are closed?
You have to jam the gears.
How did they do that?
They set a brilliant molecular trap. They knew that sodium drives the machine toward the outward-facing state. So first, they replaced the sodium in their testing environment with potassium.
Potassium is chemically similar, right?
It is, but it doesn't trigger this specific machine. So they effectively starve the transporter of its mechanical trigger.
But wait, starvation just stops it from cycling. It doesn't freeze it in the exact closed position they need for the photo.
True. For that, they used a chemical hack. They took a single amino acid near the top of the channel D262 and mutated it into a cysteine.
What does a cysteine do?
Cysteines have this unique property where they can form incredibly strong chemical bonds, disulfide bonds, with each other under the right conditions. By making this mutation, they allowed the two sides of the channel to literally cross-link and weld themselves together.
It's like shoving a steel bar into the spokes of a spinning bicycle wheel. It physically forces the transporter to freeze in that tightly sealed, occluded pose so the cameras can take the picture.
It's a classic, elegant biochemical trap. And by comparing the open pose to the frozen, closed pose, they found the core mechanism. It all comes down to one single amino acid sitting right in the middle of the elevator shaft.
Ah, the swinging cage door.
Yes, a specific glutamate residue, E284. When the transporter is open to the inside of the cell, this glutamate acts alongside the surrounding structure to form a perfect, tight molecular cage around a magnesium ion.
So it grabs the magnesium with incredibly high affinity. The magnesium is locked in.
But then our exchange student, sodium, arrives from the outside of the cell and binds to the structure.
And that binding event changes the local shape, right? It physically forces that single glutamate to pivot, to flip completely away from the magnesium ion.
Just violently swings out of position. And when it swings away, that perfect cage is broken. The grip on the magnesium is lost, the affinity plummets, and the magnesium is effectively ejected out into the extracellular space.
I love this imagery. It is quite literally a revolving door. Think of a busy hotel lobby. The person inside the lobby, that's our magnesium, steps into the revolving door. But the door is locked. They are perfectly happy standing in their little cage. They can only leave if someone from the outside, the sodium, pushes their way into the door from the street. That mechanical push forces the central pillar of the door to pivot, shoving the magnesium out onto the street so the sodium can step into the lobby.
And that tiny mechanical pivot of a single amino acid is the fundamental gear driving this massive transport cycle.
Okay, time for another quick recap. So, bullet 1, CNNM4 exchanges magnesium for sodium. Bullet 2, the researchers trap the occluded state using a clever D262C mutation. And bullet 3, the binding of sodium flips glutamate E284, releasing the magnesium.
You got it down.
So for the listener, can you visualize that single amino acid flipping? It's incredible to think that this tiny mechanical pivot dictates your entire cellular magnesium balance.
It really is mechanical perfection. But, leaves us with one glaring question. We know the tiny elevator shaft does the transport. We know the energy basement provides the fuel.
But what about that massive complex roof?
Exactly. The heavily glycosylated ECD domain sitting outside the cell, with its intricate crossed-arm communication network. How does the roof actually talk to the elevator?
Right. Because if the whole point of that asymmetric complexity was middle management, there has to be a phone line connecting the top floor to the elevator shaft.
There is. It's a literal string of amino acids called the linker. It physically connects the roof domain to the top of the elevator. But when the researchers looked at the environment this string passes through, they found something that breaks the fundamental rules of biochemistry.
I remember this part from the paper, and it is absolutely wild. Let's break down the environment first. The cell membrane, the barrier this string has to plunge into, is essentially a layer of fat. It's made of lipids.
Yeah, it is highly greasy and hydrophobic. It aggressively repels water, and it violently repels electrical charges.
So usually any protein part that sits inside the membrane is covered in neutral, greasy amino acids, so it blends in. Yet right in the middle of this buried linker string is an amino acid called arginine, residue R178.
Which carries a highly concentrated, powerful, positive charge.
But wait. Burying a strong positive charge inside a greasy, neutral lipid membrane incurs a massive energetic penalty. It's like trying to hold a beach ball underwater or forcing identical magnetic poles together. The molecule will fight with everything it has to escape that state. Why would nature design something so fundamentally unstable?
Because the instability is the whole point. By burying a positive charge in a greasy environment, nature ensures that this connection is a loaded spring.
Oh.
Yeah. The cryo-EM structure shows that this positively charged arginine only finds relief by forming a stabilizing hydrogen bond with the elevator shaft when the transporter is in the open, outward-facing state.
Okay, so when it's open, it's stable.
But when the transporter closes into the occluded state, that bond breaks and the charge is exposed to the grease again.
Oh, wow. So the roof is acting like a puppeteer.
Exactly. It uses that energetically tense, charged interaction as a literal puppet string. When the roof domain senses something in the extracellular environment and changes shape, it tugs on the linker.
Because that buried positive charge is so unstable and desperate to form its bond, the mechanical force transfers instantly to the elevator shaft.
Yes, the roof uses that spring-loaded tension to physically force the revolving door open or closed. If researchers mutate that arginine and remove its positive charge, the spring tension vanishes and transport activity drops to almost nothing.
The string puppet mechanism. All right, to quickly summarize this final piece, bullet 1, the ECD and TMD are connected by a linker. Bullet 2, arginine R178 is intentionally buried in the greasy membrane. And bullet 3, this acts as a string puppet mechanism to drive transport.
It's a masterclass in evolutionary engineering, using an energetic penalty not as a flaw, but as a feature.
So, question for you guys listening. Nature rarely makes mistakes. When you see a structural rule being broken like this, like a charge in a lipid membrane, what hidden function do you think it might be serving in other parts of your body? It really shifts how you look at architecture, doesn't it?
It really does. It shows us that tension is often the driver of function.
Absolutely. So let's step back and look at the whole picture we've built today. We started with the terrifying, murky diagnostic mysteries of Jalili syndrome and severe developmental seizures. We took a high-resolution, atom-by-atom, flash-frozen picture of the problem.
Right.
We descended through the four-story, beautifully asymmetric architecture of the human CNNM4 bouncer. We discovered the MgATP ignition key and the acidic patch gas battle. We witnessed the sodium-driven revolving door. And we saw the complex roof pulling the loaded spring puppet strings.
And, this is where the deep dive moves from just a fascinating biological blueprint into actual world-changing therapeutic potential.
How so?
Because these CNNM bouncers do not exist in isolation. They form complexes with other proteins in the cell, specifically a family called PRL proteins, and those interactions are heavily implicated in cancer progression.
Wait, how does magnesium connect to cancer?
Well, cancer cells multiply at an uncontrollable rate. To fuel that hyperactive division, they have an insatiable appetite for metabolic energy, which means they require massive concentrated amounts of magnesium. They basically hijack these transporters to feed themselves.
Oh, that makes sense. If you're building an army, you need massive supply lines.
Exactly. But now, thanks to Bai and colleagues, we finally have the precise atom-by-atom blueprint of the CNNM4 elevator, specifically locked in that closed, occluded state. And we know exactly where the acidic patch sensor sits.
Wait, I see where this is going. If we know exactly what the revolving door looks like when it's jammed, could we design a drug to replicate that jam?
That is the ultimate goal.
Right.
Could we theoretically synthesize a targeted molecule, a customized chemical stick, and intentionally shove it into the spokes of CNNM4 proteins in a tumor?
Oh my gosh.
If we can jam the revolving door and block the acidic patch sensor, we could cut off the magnesium supply line, effectively starving the cancer cells of the specific fuel they need to divide.
That is a phenomenal thought to leave on. Taking a diagnostic mystery and turning a structural blueprint into a targeted, localized weapon against cancer.
It's all about having the right map.
It really is. So as you go about your day-to-day, I want you to think about that. Think about the billions of these tiny asymmetric molecular machines firing across your cellular membranes right now. They are tasting the fluid with their acidic patch, flipping that single amino acid in the revolving door, and pulling the spring-loaded puppet strings, perfectly balancing the magnesium that keeps your brain firing and your cells alive.
It's a reminder that once we have the tools to actually see the microscopic machinery clearly, the biological landscape isn't so muddy after all. We just needed a better map.
And today, we got to read that map. Keep wondering, keep looking closer at the world around you, and we'll catch you on the next deep dive.